Welcome to Biology Made Easy. In today's lesson, we will look at the scientific method. The scientific method is the method that all scientists use, and biologists also use the scientific method. So today, the scientific method. The scientific method, therefore, we can define as a systematic, dependable, lasting method of collecting information and forming ideas based on facts. Now, science is based on facts, and these facts are obtained through the senses. Yes, the senses, we have sense of seeing, hearing, smelling, touching, and tasting. And then also the facts are also gathered by using equipments which are extension of the senses, like microscopes, thermometers, and various electronic sensors and probes. Good. Biology as a science also uses the scientific method. So therefore, let us look at the steps of the scientific method. First, observation to identify problems. 2, hypothesis or aim. 3, experimentation or investigation. Then 4, results of experiments. 5, discussion of results. Then we have 6, conclusions or inference. And then we have reporting. That's report or publish the findings. These are the various steps of the scientific method. Now let's try to quickly explain what each step means. Observation is looking at the environment. You know, as you watch the environment, you come to know there are certain things that seem to be a problem that you want to investigate. So you observe critically. Anytime you observe critically, you find something, a problem. And then we look at hypothesis. Hypothesis is a possible answer to the problem. So it's like a guess answer to the problem you found. You found a problem of animals eating your plant, and you give a guess answer that it might be snails, which are nocturnal. So that is the hypothesis. And hypothesis is what is tested in the experiment. Now let's find out about the experiment. The experiment is a setup to test the hypothesis, whether the hypothesis is true or false. That is the experiment. What happens during experimentation? During the experimentation, the following are done. One plans, you plan and assemble equipment and materials. Then you set up test and control experiments. What is a test experiment? The test experiment is the variable being investigated. We'll understand what it means soon. The variable being investigated is the test experiment. Then the control experiment is the standard or the non-variable with which you will compare the variable in the experiment. So, test experiment is the variable, something that you don't know and you have to vary. I want to find out what animals are eating these plants. I said they are snails. I suspect they are snails. That's the hypothesis. How do you find out? So you are going to look at the time and go through the farm and look at what is causing that. Meanwhile, you have another area of the farm where animals are not eating the plants. And that place is the control. You are going to see what animals are in that place. That is the standard you are comparing with the test experiment. Okay, then under the experiment, you observe to take down records. So observations are made and raw data is collected. You collect, you measure, you write what is happening. That's the raw data. The data is analyzed statistically with graphs, charts, maps, etc. Let's look at what we call analyzed data. We have raw data of the height of boys and blood groups. That's the raw data. Now, when we analyze the data, we can look at the percentage of the height, and you see that the analyzed data easily show that 25% of the boys are between the heights of 160 to 165 centimeters. All right? So that the analyzed data shows it clearly. The raw data doesn't show it. The analyzed data of the blood groups show that 40% of the boys are in the blood group O. So analyzed data makes it easy to interpret the results. So that is analyzed data. Now, after the analyzed data, the results are written out. And the results of the experiment are the written out observations and the analyzed data. Now, after the results are written, we get on to discussion of results. In the discussion of results, the results you obtain in this particular experiment is compared with results of other experiments, similar experiments that people have done. That is, I saw boys in this class, the highest percentage are within this range. Oh, other people did the same experiment and saw that all boys in class 6, where your experiment was done, have this kind of height. The maximum, the highest is boys between this. So you in discussion, you compare your results with other results. Then you have conclusions. In the conclusion or inference, you state whether the hypothesis is true and should be accepted, or the hypothesis you made is false and should be rejected. Good. Let's look at example using real experiment. Now, before you set up an experiment, you should plan. Plan before setting up experiment. And there are key things to look out for planning. One, consider the location. Where are you setting up the experiment? Is it in the lab or in the field? All right? If it's in the lab, which area of the lab? Inside, outside? If it's in the field, which area? You also have to plan considering the period or time of the experiment. Is it day or night or season, rain season or winter or what season? So the period of the experiment. And then you have to consider plan before the type of equipment and material to use as control and test experiments. And what to do just before the experiment is to plan to set up a model of your experiment. Set up a model or sketch the plan of your experiment on paper. And then prepare record sheets to record your measurements. Now, we are going to use the experiment a student did to look at these facts that we have discussed. Let's look at the student's problem, the student's investigation. A school started serving 3 types of milkshakes for breakfast. A student who is allergic to juice of fermented corn felt sick repeatedly each time after drinking the milkshakes. The student suspected that the milkshakes have juice of fermented corn in it. The student set up an experiment. Good. So the student had some milkshakes served in the school. 3 types of milkshakes, which we have labeled A, B, and C. And the student found out that anytime he had the milkshakes, the student got sick. So the student decided to find out what is it in this milkshakes that makes me feel sick. Because the student knew that anytime he has the juice of fermented corn, he feels sick. So he thought that there's some juice of fermented corn in one or all the milkshakes. So please note any wrong steps or facts the students will go through. And at the end, we will discuss. So write down the observation of the students. Became sick after taking milkshakes. Hypothesis. This is the hypothesis the student stated. It's likely that one or all the milkshakes contain juice of fermented corn. Yes, so the student planned the experiment as a sketch. He sketched it instead of building a model. So the sketch, the student sketched out. These are the 3 milkshakes. So this is milkshake A, milkshake B, milkshake B. These are the milkshakes. And then decided that he will have a test tube rag and put test tube rag and fix test tubes in it. And then so this was the plan. So some milkshakes in this one, A1, that's milkshake A, B, B1, C, C1, X, X1. And this is the control. And these are the milkshakes. So the student had this plan, and the student listed all the equipments and the materials he will need. Yes, so the student has his plan of the milkshakes, raw milkshakes in this, and then and these test tubes, and he had his measuring cylinders and other glassware listed as we have listed them. Then the student decided to do a preliminary experiment to find out which substance he can use for his control experiment. And the student decided that juice of fermented corn that he is allergic to has starch in it. So he can either use starch as the control experiment or the fermented juice. So he decided that, well, let me test if there's really starch in the fermented juice. Yes, so the student did this preliminary test to find out whether there's starch in the corn juice, whether he should use the corn juice as a control experiment or starch itself. So put 5 ml of starch in here, put some 3 drops of iodine solution, and you know iodine solution is brown in color and turns blue-black in the presence of starch. So the starch solution, of course, showed the starch, and the juice of the corn also showed that it has starch. So the control experiment has the fermented corn juice, And so the student put 5 ml of the corn juice to 5 ml into X. Good. Yes, so the student measured 5 ml of each of the milkshakes into the test tube. 5 ml of each of the milkshakes. So 5 ml in A, 5 ml in A1, that's A. A1 is a spare. 5 ml in B1 and B. 5 ml in C and C1. All right? Then tested for starch in each of these. So A, 3 drops of iodine solution. Iodine solution is brown in color. So 3 drops of iodine solution went into each of the test experiments. Look at the control experiment. Iodine solution turned blue-black. Then we have A, milkshake A. The iodine solution remains brown. Brown color of the iodine solution remained. B, the iodine color of the iodine solution remained. It's still brown. Well, milkshake B. Milkshake B1, the iodine solution remains brown. And milkshake C, the iodine solution became blue-black. So therefore, there's starch in milkshake C. Let's look at how the students set up to write the results and the conclusions. Milkshake A remained brown with iodine solution, which indicates that there is no starch in milkshake A. Milkshake B remained brown with iodine solution, which indicates there's no starch in milkshake B. Milkshake C turned from brown to blue-black when iodine solution was added, indicates that there is starch in milkshake C. Then the students went on to say that there is juice of fermented corn in milkshake C because the iodine solution turned from brown to blue-black in milkshake C. Then in the discussion of the results, the students wrote, iodine solution turns brown to blue-black in the presence of starch. This agreed with what happened in the starch in the juice of the fermented corn in milkshake C. So therefore, conclusion, milkshake C contained juice of fermented corn. Now, questions. Write down the good procedures of the students. 2, write down wrong facts the student stated and suggest how the facts can be corrected. this is the end of today's experiment. We tried to sort out the scientific method. That's the method or the way scientists work. We discussed the scientific method, and we discussed an experiment a student made, the procedures the student made, his hypothesis, and how the hypothesis was tested in the experiment. Because hypothesis is the guess answer and is tested in the experiment. He wrote out his conclusions. And what do we expect to have in our next lesson? In our next lesson, we are looking at something very different. We are going to look at diversity of organisms. And we are going to look at the cells as prokaryotic cells, eukaryotic cells, and look at eukaryotes, viruses, in our next lesson. So till we get together again in our next lesson, thank you and goodbye.